Good morning, everyone. Welcome to this lecture, another lecture in the series of molecular biology. Though we had discussed multiple aspects of molecular biology in our previous lectures, today we'll be focusing on a very relevant topic which lays the foundation of understanding the operant concept in prokaryotes, or which laid the foundation of multiple screening methods that are currently being used, multiple vectors that are currently being used in recombinant DNA technology. And yes, my dear students, we are talking about Lac Operon today. So, what is Lac Operon? What is Operon concept? And that will be our main aim for this particular lecture. We should be able to have a clear-cut understanding of what is Operon concept, what is Lac Operon, what are the proteins that are being coated by the Lac Operon, how it is being regulated by different sensing sensory parts, how the choice in terms of bacterial choice that we are talking about, the choice of E. coli in terms of its substrate liking, whether it should go for glucose or it should go for lactose as such. What what pathway or what molecule actually recognizes the variation in the concentration of glucose and lactose, and how this particular lac operon is switched off and on, and how the expression of this protein, the expression of the proteins is happening. Then we'll be studying different mutants also, that yes, mutation in this region can change the phenotype of the E. coli. So, in nutshell, the major outcome of this particular lecture will be, you will be having a clear-cut understanding. You will be having a clear-cut understanding that what exactly is the function of Lac Operon, how the regulation is happening, and if we are talking about certain critical aspects, that what will happen if we mutate a certain gene, what would be the change in that particular Lac Operon, whether it will be switched on or off, and that complete understanding will be established in this particular lecture. So, without any delay, let us first focus on what is Operon. What do you mean by Operon concept? Of course, the Nobel Prize winners Jacob Monod and Wolf, Lof, and they are credited for the discovery of this operon concept. They first of all gave the lac operon in E. coli, and for that they were awarded with the Nobel Prize. But we have to clearly understand first of all what is operon concept. See, when we are talking about eukaryotes, we have exons, introns, and exons. That means we have certain coding regions, and in between we have certain non-coding regions. And later on, at the stage of mRNA splicing, all the exonic regions are coming together to give rise to amino acid chain, to give rise to a polypeptide chain, which is further folded to give rise to a conformationally active three-dimensional protein structure. But in prokaryotes, there is no intron. There are no introns. There is no non-coding region. So what is happening? All the genes which are involved in a single pathway are clustered together, and that gene cluster is involved in a single pathway is called as operon, and that's how we define it. So what we are saying, genes which are actually coding for enzymes, genes that are coding for enzymes, and those enzymes are related to a single metabolic pathway. So all that genes are organized into a single cluster, and that cluster within the genome is called as operon. That cluster within the genome is called as operon, and it is a common sense scenario that when we're talking about the word operon, we are specifically talking about the prokaryotes because it's not present in the eukaryotes. So when we are talking about the array arrangement, there's the multiple genes are present in a short cluster, and that cluster is called as operon. Then that cluster is also called as polycistronic part. Polycistronic part. So you see there are multiple genes, and each gene is present as a cistron, and there is these cistrons are present adjacent to each other to give rise to a polycistronic structure. So main thing that we should remember, that all these genes that are involved in a single pathway of the polycistronic is governed by a single promoter region, which is upstream to these particular coding sequences from the start site, and that single promoter is the regulator point along with the operator to control the downstream genes, to control the expression of the downstream genes. So when we are talking about the regulatory elements, there are two types of regulatory elements that we usually come across in the molecular biology. One first is cis acting element, and the other one is trans acting element. So although it needs no introduction, that cis acting elements are the regulatory regions that are present on the DNA where it influences the expression of the genes that are present in the operon, and they are present in the immediate vicinity. They are simply few bases away from the upstream region. From the start site, they are just few bases away. Whereas the transactive elements, these are the molecules that actually will interact with the cis acting element to cause the effect on to regulate the expression of these polycystonic genes. So what is happening? What is happening? See, there is a polycystonic part, and then there is a promoter. Fine. Then these elements or promoter are cis acting elements because they might be regulating the expression of the gene. But there is far away, there is a certain gene whose product is actually governing the expression of the gene which is present here. So that element is called as transacting element. So when we are talking about specific Lac Operon, it was given by Jacob and Monod in 1959. 1959. So you see, at that time only they discovered this particular concept. They discovered this particular concept, and what they showed us, they showed us that there are three genes which are present in E. coli, which actually permits the bacteria to use lactose as an energy source, to use lactose as a sugar for energy source. So the genes which are present here are LEC-Z, Y, and A. LEC-Z, LEC-Y, and A, which codes for LEC-Z codes for beta-galactosidase, LEC-Y codes for galactoside permease, whereas the LEC-A codes for galactoside transacetylase. So when we are talking about the principal enzyme which actually uses lactose and metabolizes the lactose, that is beta-galactosidase. It breaks down actually lactose into its monomeric form of galactose and glucose. Whereas permease actually allows the entry of the lactose inside the cell. And transacetylase, what it does, after the metabolism of lactose, the byproducts which are accumulated as a toxic form, it is responsible in detoxifying them, so that there is no toxicity of the lactose metabolism. So you see, all the three genes are present in a single fashion. So these genes, LEC-Z, LEC-Y, and LEC-A, are grouped together to form a gene cluster, a three-gene polycystonic region, a three-gene polycystonic region. And please remember, when we are talking about the Lac Operon here, please remember the important part. What is the important part? See, in order to form a protein or the enzyme beta-galactosidase, permease, and transacetylase, ribosomes are required to bind on the mRNA which will be made from this particular DNA. Now suppose an mRNA is formed here. Then individual cistron holds an ribosome binding site, So the ribosome can come and bind to the mRNA, which is a single mRNA, bind to the first gene, then another ribosome can bind to the second gene, and the other ribosome can bind to the third gene. So there is a simultaneous independent expression of beta-galactosidase, transacetylase, and permease. So there is no single polypeptide chain as you see in the cases of virus. They require the protease to cleave the polypeptide chain to give rise to functional protein. But despite of a single gene cluster, despite of the single cluster of multiple genes in prokaryotes, the expression of a gene is happening because they contain the sites, each cistronic part, each cistron contains the binding site of the ribosome, so that they give rise to a functional protein. Now this is the representation, this is the simplest representation of Lac Operon. We have structural genes, that is like Z, like Y, like A. Upstream to the start codon, we have a promoter and an operator. We have a promoter and an operator sequence. This promoter sequence, the P region, the promoter sequence is the site where the RNA polymerase will be binding, and then that RNA polymerase will be forming an open complex, and then it will be transcribing the DNA to give rise to an mRNA. And far away at a transacting position, there is a small gene called as repressor gene, which is denoted by LEC-I. I stands for inhibitor. And collectively, this is called as Lac Operon. So how it functions, how this control of Lac Operon is happening. So the control is happening in two different types. One is the negative control, and the other is the positive control. What is negative control? Negative control is applying the brake, right? What you are doing, you are switching off the expression of Lac Operon. There will be no expression of LEC-Z, Y, and A. Whereas the positive control, it potentiates the expression of Z, Y, and A. So what it is doing, it is actually stimulating the transcription of Lac Operon. It is actually stimulating the transcription of Lac Operon. And when it is happening, It happens only and only when the glucose concentration depletes, when the glucose concentration is finished. So what it is happening? Let us go one by one to these aspects, and let us first discuss the negative control of Lac Operon, that how it is happening. See, although what we have understood the basic structure of the Lac Operon. Let us see what exactly is happening. Under normal condition, if I say LAC-I gene, this LAC-I gene actually codes for a repressor protein. Now it will express itself under normal condition, and then the repressor protein which is formed, the 4 units of the repressor proteins will be forming a tetramer, and that tetramer will be going and bind to the operator region in such a manner that it overlaps with the promoter region also. So you see, the inhibitor protein or the repressor protein in tetrameric form is going and binding to the operator, and this binding results in inhibition of the binding of RNA polymerase to the promoter site. So in the presence of repressor protein in a bound state at operator site, RNA polymerase is not able to bind to the promoter. Thereby, there will be no transcription and expression of Z, Y, and A. So the complete Lac Operon is switched off. So the complete Lac Operon is switched off, and this is called as negative control. This is called as negative control. Whereas when we are talking about the inducers of the Lac Operon, that inducer is none other than allolactose. The inducer is none other than allolactose, which is an alternative form of lactose. What it is doing. Now, this particular allolactose or the lactose will go and bind to the repressor protein. It is not binding to the same catalytic site where the operator is bound, but in fact it is binding to another allosteric site, which after binding it will change the conformation of this particular repressor protein in a way that now it is not able to bind to the operator. So what is happening in presence of allolactose? The repressor protein changes its conformation and binds, and because the allolactose or the lactose is now bound to the repressor protein, the complete complex is not able to bind to the operator. So you see, now the region is free. Now there is free access to RNA polymerase, and RNA polymerase can relish the free access and can bind to the promoter, thereby causing the transcription and the expression of Z, Y, and A, and in turn switching on the Lac Operon. So this is the negative and positive control. This is the negative and positive control that is happening. But one has to understand when actually negative control is happening, when actually positive control is happening. So depending upon the environment of the E. coli or the bacteria, what we are talking about, this Lac Operon is in either off mode or in on mode. In normal conditions, when we are talking about sugar as an energy source, the preferred source is always glucose, right? Because it's a monosaccharide, it is readily consumed. Whereas when we are talking about the lactose, first of all, the E. coli will be making efforts to break down the disaccharide into monosaccharide, and then only lactose will be utilized. So the bacterium always prefers glucose over lactose. That is the bottom line. That if both the sugars will be present, then the liking or the E. coli will be first eating up the glucose, and then it will focus on lactose. So when we say, based on this particular aspect, what can be the fate of the Lac Operon in terms of on and off? So let us see. When glucose is present and lactose is absent, then there is no point of expressing Z, Y, and A. So always it is switched off. When we are talking about that glucose is present and also lactose is present, both are present, then what happens? Then also E. coli will prefer first to take glucose, and it is not able to consume lactose. Why? That we'll see in coming slides. But preferably it always takes glucose. So the Lac Operon is switched off. And when both are absent, then also there is no point of switching on the lactose. But when glucose is vanishing, the glucose vanishes, glucose is absent, and only lactose is present, then and then only E. coli will switch off the Lac Operon in order to survive and in order to metabolize this sugar known as lactose. You see, that is the possible fate. Now, when we are talking about that lactose is absent, when lactose is absent and when only glucose is present, then what is happening? The Lac Operon is switched off, and that is the function of your repressor protein. The repressor protein will be in bound state to the operator. The repressor protein will always be in the bound state to the operator when glucose is in excess, glucose is in ample amount. And when lactose is present, then what will happen? A small amount of allolactose will go and bind to the repressor protein, as I narrated you, on the allosteric side, the side which is not the part of the catalytic side. After binding, it will induce the conformation change in the repressor protein, thereby releasing it from the operator and promoter region. Now there is free access to RNA polymerase, which can bind to the promoter site and cause the expression of ZYNA, which is shown here. There is a conformation change due to which the dissociation has happened of the repressor protein, and RNA polymerase can now come and perform its function. Now, more interesting part for me is when both the sugars are present, then what is the mechanism that the Lac Operon is off? Because lactose is present, it can still bind to the repressor and can switch on the Lac Operon. But as I said, that even when lactose and glucose both are present, the Lac Operon is switched off. Lac Operon is switched off. How? See, let me tell you, when we are talking about that both are present, then due to the presence of lactose, the allolactose will bind to the repressor protein and change its conformation, thereby it will remove it from the operator. That's the normal process that we did. And because there is a free access, RNA polymerase should come and bind to the promoter, thereby causing the transcription process. But what happens? RNA polymerase comes and binds to the promoter region and it falls off. It starts binding again and then again it falls off. It is not able to form an open complex for transcribing the ZY and A. Why? Because it is unstable. The binding is unstable. They require an additional protein to stabilize this particular binding. And that particular protein is not expressed until the glucose is finished. So you see, even if the lactose is present simultaneously with glucose, there is no necessary protein to stabilize the binding of RNA polymerase to the promoter region of Lac Operon. That is the reason. That despite of lactose presence, the Lac Operon is switched off. And when we are talking about that glucose is now finished, glucose is now absent, and only lactose is present. Now, E. coli requires lactose. E. coli requires the energy, so it will break down the lactose. So for that particular purpose, Lac Operon is required to be switched on. So in presence of lactose, of course, the repressor protein is now changed. Now there is a free access of promoter to RNA polymerase. RNA polymerase will come, and simultaneously an activator protein will be coming and stabilizing the binding to promoter region and then only the open complex will be formed and the transcription process will start. So the expression of ZYAF will happen. Now let's summarize what we have read till now. So what we are talking about? We are talking about that if glucose is present along with lactose, that is showing that RNA polymerase will be able to bind to the promoter, but will not be able to transcribe. That means the Lac Operon is switched off. The promoter will keep falling from the site. Okay. When we are talking about glucose is present but lactose is absent, then also the complete Lac Operon is switched off. Why? Because RNA polymerase binding to the promoter is blocked by the repressor protein. When both are absent, there is no transcription. Why? Because the complete thing is blocked by the repressor. But when only lactose is present, then the transcription is happening. Why? Because RNA polymerase is having the access to the promoter site and it never falls off. Why? Because an activator protein is also present, which is stabilizing this particular binding. So The next question comes. You keep on talking about activator protein, but please tell us what is the activator protein? And that activator protein complex is none other than the defining positive control of Lac Operon, which is mediated by none other than cyclic AMP and catabolite activator protein. So what is happening? What is happening? Who senses that glucose concentration is finished? There should be some sensory mechanism which should recognize that glucose has depleted. Now we have to switch on the Lac Operon. See, the molecule what we are talking about, that is cyclic AMP, when the glucose concentrations start lowering and then reaches to the almost finishing level, the concentration of a nucleotide that we are calling as cyclic AMP, it is nothing than cyclic adenosine monophosphate. This is a nucleotide which was triphosphate, but the terminal pyrophosphate is released, and the first alpha phosphate forms a bond with the 3' OH, with the 3' hydroxyl group to form a phosphodiester bond, and thereby cycling. Thereby cyclization has occurred. So that's why we are calling it as cyclic AMP. And this cyclic AMP is acting as secondary messenger in multiple signaling pathway. It acts as secondary messenger. So when we are talking about depletion in the glucose concentration, simultaneously the concentration of this particular nucleotide called as cyclic AMP rises. So that rise in cyclic AMP concentration is the indicator that the substrate of glucose has now depleted. Now this cyclic AMP should result in activation of Lac Operon. But how? But how? Even in the lab scenario, when we are talking about the experimental part, if you grow an E. coli culture, and you want to overcome the repression, you want to overcome the catabolite repression, and you want to initiate or start the Lac Operon, what people do? They add cyclic AMP. Even in that condition where the glucose concentration is okay, it is there, it is not depleted. If you add high concentrations of cyclic AMP from outside, you will see that there is a reversal of catabolite repression and the Lac Operon starts. So you see, the addition of cyclic AMP, what it is doing, it is leading to the activation of Lac genes, the Z, Y, and A, even in the presence of glucose. So that is the crucial part, which actually dictates the positive control of Lac Operon. Now, when we are talking about the positive control, there are two controllers. First is cyclic AMP that we had discussed. Another one is CAP, catabolite activator protein, called as CAP. Together, this particular cyclic AMP and CAP, they both form a complex, and then that complex is required for positive regulation of Lac Operon. This particular CAP protein that we are calling as catabolite activator protein, is also called as cyclic AMP receptor protein, or CRP, and that particular CRP is coded by a gene, that is a PRIP gene or CRP gene. So what is happening? The complex between the the catabolite activator protein, and cyclic AMP, it goes and binds to the Lac promoter. When it goes and binds to the activator site of the Lac promoter, it potentiates the binding of RNA polymerase to the promoter. So when I was saying that an activator protein will stabilize the binding of RNA polymerase to its promoter sequence, that particular activator protein is none other than the complex of cyclic AMP and CAP. So when suppose you're going for mutation based studies and you have you have a mutant whose Lac gene is not stimulated by this particular complex. There is a mutation in the promoter. There is a mutation in the activator site of a promoter, but not on the site where RNA polymerase is binding. This mutation will specifically inhibit the binding of this particular complex on promoter. So what will happen? If this binding is inhibited, it was observed in the lab that Lac Operon was not able to start because RNA polymerase was continuously binding and falling off. So it was concluded that this particular complex is of utmost importance for RNA polymerase binding to the promoter and their stability. And that is the beauty of CAP protein. Even not only Lac Operon, this binding site of this particular CAP protein, the catabolite activator protein, is present in Lac Operon, it is present in lactose operon, it is present in arabinose operon. But you will be very surprised to see that the binding site of this cap protein is highly conserved in these operons. The binding site will always contain the sequence of PGTGA, and this sequence conservation indicates one important role. And what is that? That is the binding of cap protein should be conserved when we are talking about switching on and operon. Why? Because this specificity will induce a higher affinity and tight binding to the DNA. So when we are talking about, apart from this tight binding, if we talk about the promoter region which is present here, whether that promoter is strong promoter or weak promoter. So please remember, the promoter which is present upstream to the Lac Operon is always a weak promoter. Why? Because if it would have been a strong promoter, then what could happen? Even in the glucose presence, the Lac Operon will always be on. That is the characteristic of a strong promoter, right? But what we have observed, that in the presence of glucose, that particular RNA polymerase is not able to get stabilized on the promoter. This is the indication that the promoter is weak. It requires an additional factor to stabilize it. Whereas if it would have been a strong promoter, there would be no requirement of any kind of extra activator protein to stabilize the binding of RNA polymerase to the promoter region. So, what is happening when we are talking about the positive control? Actually, the catabolite activator protein is binding to the site of promoter called as activator site, and it is facilitating the binding of RNA polymerase to the promoter. Thereby, after binding and stabilization, this particular RNA polymerase will be able to form an open complex which is required to transcribe these particular three genes called as LEX-Z, Y, and A. LEX-Z, Y, and A. And please remember, form of cap this form of cap exists only and only when the glucose concentration is absent. When the glucose concentration is absent. So this is how the complete positive control is there. But suppose the glucose concentration is again there, or the glucose concentration is there, it is not depleted, then what will happen? What will happen? The shape of the cap protein is changed whenever there is a presence of glucose. The conformational shape of the cap protein, the catabolite activator protein, is completely changed until and unless the cyclic AMP concentration rises and the cyclic AMP is bound to the catabolite activator protein to give rise to a complex. This conformational shape will not be sufficient enough to bind to the promoter. Once the glucose is depleted and the cyclic AMP is bound, then the shape changes, and that change in shape increases the binding or initiates the binding to the activator site of the protein, activator site of the promoter, and thereby switching Lac Operon on. So this is how the complete positive control is going about. So you see, when glucose is present, Lac Operon is always in relatively inactive state. So when we are talking about favoring the glucose, we are talking about that the complete Lac Operon will be in switched off condition, and that particular condition is called as catabolite depression. That particular condition is called as catabolite depression. Why? Because it is using a breakdown product to depress your operon. It is using a breakdown product to depress your operon. That's why it is called as catabolite depression. So when we are talking about the regulation of operon, till now what we have read, the transcription regulation is managed by when it is in off condition. When only lactose is absent. When lactose is absent, the complete operon will be repressed. But when lactose is present as well as glucose, there is a de-repressed condition. Right? When Why it is calling as depressed condition? Simply because repressor protein is detached from the operator region and RNA polymerase is binding. Although it is falling off, but initial process is there, it is binding to the promoter region. That's why we're calling it as de-repressed state. It is active but not able to transcribe. And then it becomes really active when lactose is present and glucose is absent. The positive activator is coming, and it is inhibiting the catabolite depression. The RNA is stable, RNA polymerase binding to the promoter is now stabilized, and like Z, like Y, and like A expression is happening. So that is actually the complete aspect of your operon system. So let me ask you a certain question. After understanding all this particular Lac Operon concept, I am asking you four sets of questions and you have to suggest the phenotype of Lac Operon. What will happen if Lac Operon? What will happen if these particular conditions are happening? What will happen to the fate of Lac Operon? Suppose uh, I have a mutant of Lac I negative. If I have Lac I negative, what will be the condition? Whether whether the operon will be on or operon will be off. Whether the operon will be on or the operon will be off. What will be the fate? See, we are talking about the Lac I. Lac I is leading to the formation of repressor proteins which are in tetrameric form. These repressor proteins will be going and binding to the operator region. Where is the operator region? Just adjacent to the promoter. Thereby, thereby they will be inhibiting the binding of this particular RNA polymerase. This binding now is inhibited. Why? Because there is an additional repressor proteins which have occupied this particular space, and there will be no downstream processing of ZY and A. There will be no downstream expression of this ZY and A. That is happening, of course, under normal condition. But what will happen if this Lac I is mutated? If this Lac I is mutated, there will be no formation of repressor protein. And if there is no formation of repressor protein, then there is a free access of RNA polymerase to promoter and operator. This RNA polymerase will go and bind to the promoter and operator, and in presence of positive regulation, it will cause the transcription of this ZY and A. So the Lac Operon is now on. The Lac Operon is now on. That will be the phenotype. Now suppose, suppose what I say. Listen carefully. Suppose the promoter region is mutated. The promoter region is mutated. What will happen? If the promoter region is mutated, then there will be different things. If promoter is mutated at activator site, then the positive regulation, will not be happening, and the RNA polymerase will be binding and falling off. So the phenotype will always be switched off. If the promoter mutation is happening on RNA polymerase binding site, then what will happen? No matter your positive control is there, doesn't matter your lactose is there, doesn't matter your glucose is depleted, the polymerase will not be able to bind to the promoter. Thereby, there will be no switching on of your Lac Operon. So the Lac Operon will be switched off always. Now another aspect, let us see what will happen. If the operator is mutated? What will happen if the operator is mutated? It's very simple. See, if the operator region is mutated, this particular repressor protein which is now formed will not be able to bind to this particular site, because this particular site is now defective. So if it is not able to bind, the RNA polymerase will gain complete access again for the promoter and operator, and then transcribing ZY and A. And that is the reason that I am saying that operator mutation will render the switching on of Lac Operon. So you see, based on mutation studies, there are different phenotypes. There are different results for this particular Lac Operon. So we have to critically understand that what will happen. Suppose when I ask you another question, that what will happen? If we have Lac Y negative? That means the complete Lac Operon has a mutated Lac Y gene. If this particular gene is mutated, then what will happen? What is happening? We will be having beta galactoside which is active. We will be having trans-acetylase which is active, but we will not be having any activated permease. If this permease is not there, then who will allow the entry of lactose from outside to inside? And if lactose is not able to enter inside, then there will be no one to remove this particular repressor proteins which are bound here, because that is the job of an inducer, and under normal condition that inducer is allolactose, and if that allolactose is not able to enter from outside to inside, which was the function of permease. Now since lac Y is mutated, permease is not active, lactose will not enter inside the E. coli, thereby the complete lac operon will be again in off condition, switched off. Switched off, there is no inducer to induce, even if the glucose is depleted. And even you go for complete glucose absence, if like Y is mutated, then there is no switching off. So no switching on of leg. Now next very important aspect. What will happen if leg Z is mutated? Leg Z is negative, it is mutated. If there is a mutation in leg Z, what it is showing? It is showing that your principal enzyme, which is required to metabolize or to break down the lactose into its monomeric galactose and glucose, it is mutated. It will not be formed. So beta galactosidase, beta gal, what we say in our genetic engineering aspect, that beta galactosidase, beta gal is now mutated. So although the operon will be switched on, but there will be no utilization of lactose. The operon, yes, it will be switched on. The operon will be switched on, but there will be no utilization of lactose. There will be no utilization of lactose. And this particular aspect of beta galactosidase is utilized in recombinant DNA technology in order to establish the blue and white screening method. So if you are interested in understanding that blue and white screening method, you should go to a recombinant DNA technology book and understand how this particular beta galactosidase expression is utilized, or how this particular leg Z gene is utilized as a marker, as a selection marker in terms of genetic recombinant, in terms of genetic engineering. See, with the substrate of X-gal, if you give the substrate with the name of X-gal, this beta galactosidase will metabolize the substrate and will give you blue color bacterial colonies. So that is a marker. That is a marker of LEX-Z expression. And for inducer, you can use different inducers. We are talking about the about a natural inducer, that is lactose. There are artificial inducer also. There is a very potent inducer that is called as gratuitous inducer. Gratuitous inducers. What are gratuitous inducers? Gratuitous inducers are the inducers which are actually inducing the operon, but themselves they are not metabolizing. There will be no effect on their concentration, but yes, they will definitely switch on the lac operon. And one such type of gratuitous inducer is IPTG, IPTG, isopropyl thioglycoside, and this is used in regular settings in lab to induce to switch on this particular lac operon. So that is what I wanted to narrate you before going to the end of this particular lecture, and that is what we have established the understanding based on our learning in this lecture, that at the end, if you mutate each and every aspect of each and every player of that lac operon, what will be the phenotype and what will be the resultant appearance, whether it will be on or off. So students, that is all from my side with the topic with the short topic of lac operon. Although I can still go for an hour with this lac operon, but yes, we have to summarize it here. And be just to ignite your curiosity. See, curiosity is the force that drives us to learn more and more, to understand more and more. And for your further readings, I have incorporated these particular links so that you can utilize them in better understanding. So these are the MCQs which you will be thoroughly enjoying once you go through the complete lecture. That what is lac operon is an example of what type of regulation in the presence of lactose. How long does it take for the lac operon to be expressed? How long it takes? As long as lactose is more than glucose concentration, or as long as lactose is more than galactose, or when glucose is more than lactose. So these kind of questions always appear in terms of any competitive exams, only competitive exams. So what will be the sequence of structural genes? What is regulation of lac operon can be mentioned by regulation of which enzyme, that is enzyme. Then when it will be turned on or off, or in a cell, as per the operon concept, the regulator gene governs what kind of chemical reaction. Or the gene product of Lac A, the last one. What is that? What is that? The condition in which Lac Operon is being transcribed at very high level, being transcribed at very high level. What is that condition? What is that condition? You will see here. There is written, the absence word is written nowhere. We are talking about in terms of high and low. So I'm not saying it's a glucose starved condition, it's a glucose depleted condition here. So you have to first read the lecture thoroughly, and then you have to answer all these questions and what is the correct options from it. So this is all from my end. This is the narration that I wanted to give you regarding the Lac Operon. I hope you will thoroughly enjoy this particular lecture, and you will be gaining much fruitful information in terms of understanding the Lac Operon and solving these types of MCQ. So with this, I thank you for your kind attention, and I hope that it was a happy learning hour for you. So we'll meet again in another lecture with some another topic in the series of molecular biology. Till then, please stay safe and wish you a very, very happy learning. Thank you so much.